This is the second part of Unit 2 in microbiology. Eukaryotic cells have some similarities and some differences from prokaryotic cells. The glycocalyces in the eukaryotes is present in animal but not in plant cells. It is sticky, helps to anchor the cells, and provides strength and protection. Flagella also differ in eukaryotes. They are composed of chains of tubulin to form microtubules. They have 9 pairs of microtubules surrounding 2 microtubules in the filament for what is referred to as a 9+2 filament. There is no hook, and the basal body has triplets of microtubules with no microtubules in the center for what is known as a 9+0 basal body. Cilia are not present in prokaryotes, only in eukaryotes. These are a hair-like structure around the cell with 9+2 shafts and 9+0 basal bodies. We find some other differences between the prokaryotes and eukaryotes. The cytoplasmic membrane is a fluid mosaic like in prokaryotes, but also contains sterols to help maintain the fluidity of the membrane. Both will control movement in and out of the cell, but there is no group translocation in the eukaryotes. Instead, we see a process known as endocytosis. In endocytosis, the cell uses its membrane to engulf a particle instead by manipulating itself around using the cytoskeleton. The reverse process is exocytosis, used to export a substance. Here is a brief video on endocytosis.
Cells also use energy to transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior. In the opposite process, called exocytosis, the sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents.
Ribosomes are larger in eukaryotes, being 80S with a 60S and 40S subunits. Ribosomes may also be attached to the endoplasmic reticulum. The eukaryotic cytoskeleton is more extensive with microtubules made of tubulin, smaller microfilaments of actin, and intermediate filaments made of various proteins. The centrioles lie at right angles near the centrosome. The centrioles have a 9+0 arrangement. Their function is not fully understood, but they are part of cytokinesis and form the cilia and flagella. The nucleus can be single or multiple nuclei. Most of the genetic information is contained in the nucleoplasm. The nucleolus or nucleoli is the one or more regions where the RNA and ribosomes are synthesized. Inside also has the chromatin, which is a thread-like mass of the DNA. It contains proteins called histones for packaging the nuclear DNA. The nuclear envelope is a double membrane of phospholipid bilayer. The second membrane is thought to be the result of originating as an engulfed cell, keeping its original membrane and the one put around it when it was engulfed. It has nucleopores to control the import and export of substances into the nucleus. The ER, or endoplasmic reticulum, is continuous with the outer membrane of the nuclear membrane. It is arranged as hollow tubules for transport and packaging. The smooth ER does not have ribosomes and is involved in lipid synthesis and transport. The rough ER has surface ribosomes. Protein made by the ribosomes are inserted into the lumen of the ER and transported. The Golgi apparatus receives, processes, and packages large molecules for export from the cell. Secretory vesicles are secretions which fuse with the cytoplasmic membrane to do exocytosis and push out the substance. Vesicles or vacuoles are large sacs used to store and transfer chemicals. Large vacuoles are found in plants and algae to store starch, lipids, and other substances. Lysosomes in animal cells contain catabolic enzymes. Peroxisomes contain hydrogen peroxide, catalase, oxidase, and many other enzymes used by the cells. Mitochondria are the organelles for ATP production. Inside is the cristae, or inner membrane, made of two phospholipid bilayers. The inner matrix contains the small prokaryotic 70S ribosomes and circular DNA. Chloroplasts are the light-harvesting organelle in photosynthetic eukaryotes. They have two phospholipid bilayer membranes and 70S ribosomes. The photosynthetic prokaryotes have photosynthetic lamellae instead. The endosymbiont theory is an explanation for the development of the eukaryotic cell. It is believed the eukaryotic cell formed from phagocytosis of smaller aerobic prokaryotes by larger anaerobic prokaryotes. The smaller prokaryote became the internal parasite, which lost the ability to exist independently, but retained a portion of the DNA, ribosomes, and cytoplasmic membrane. This explains the presence of the double membrane and the 70S ribosomes. And here is an animation on the endosymbiont theory.
The eukaryotic internal membrane system, called the endoplasmic reticulum, and the nuclear envelope may have evolved from infoldings of the plasma membrane in an ancestral prokaryotic cell. Such infoldings are common in modern prokaryotic cells. The theory of endosymbiosis proposes that a critical stage in the evolution of eukaryotic cells involved endosymbiotic relationships with prokaryotic organisms. Microorganisms that live within other cells and perform specific functions for their host cells are called endosymbionts. According to the theory, energy-producing bacteria may have been engulfed by a larger primitive cell and come to reside within it, eventually evolving into what we now know as mitochondria. Photosynthetic bacteria use photosynthetic pigments embedded in internal membranes to derive energy from sunlight. These bacteria may have come to live within early eukaryotic cells, leading to the evolution of chloroplasts. Several facts provide evidence for the endosymbiotic hypothesis. A few examples include: 1. Mitochondria and chloroplasts contain their own circular DNA, similar to DNA in bacteria. 2. Both mitochondria and chloroplasts are surrounded by two membranes. The inner membrane is probably evolved from the plasma membrane of the engulfed prokaryote, while the outer membrane is probably derived from the cell membrane of the host. 3. Mitochondria are about the same size as bacteria. 4. Mitochondria appear to have been derived from purple bacteria, and chloroplasts derived from photosynthetic bacteria.
So again, this link is available to you outside of this presentation.